Good afternoon. I want to thank all the presenters from this morning. I'm actually in awe on the patterns that were shown. They very much match up with what I'm about to show you here. So I would like to start with thanking my coworkers. This is truly a team effort, and we've worked together for a long time to bring you these data. So monitoring marine benthic communities has revealed changes in species abundance and species diversity in the Sound over the last 20 years. And that the chemicals measured do not correlate well with our benthic communities. They're not telling us that pollution is an issue at this point. They don't show the community distribution either spatially or temporally. And laboratory toxicity tests conducted are not giving us the answers either. They may be pointing to changes in biogeochemistry rather than in pollution patterns. Why do we monitor benthos? Benthos contribute substantially to the food web, both the benthic and the pelagic food web. Benthic taxa contribute about 21% of the biomass to the zooplankton community, which provide an important food source for fish like herring and salmon, and then obviously transferring up the food chain. Benthos regulate biogeochemical processes. By burrowing through the sediment, building their homes and their feeding activities, the benthos, they mix up the sediment, bringing low oxygenated sediments to the surface. Bioturbation helps remineralize the water column with nutrients in this way. Mixing it up, they bring bottom sediments up and making it available for nutrients to be exchanged into the water column. And some benthic species have a tremendous commercial value as well as being a traditional value. Things like clams, salmon, gooey duck, and even sea cucumbers represent a big commercial value. Assessing the condition of the benthos is done by the Washington State Department's Marine Monitoring Unit. And we assess the benthos with several programs. One is our long-term monitoring program. We visit 10 sites, and we have done that since 1989. And another program is our regional program, where we transfer through the Sound through 8 different regions and evaluate that every 10 years. So we get a spatial characterization every 10 years of the whole Sound. Along with the benthos, we collect physical characteristics, chemical pollutants, and laboratory toxicity tests. So over the last 20 years of the regional assessment of Puget Sound, we have seen declines both in benthos abundance and benthos diversity. The map on the left shows 5 sediment monitoring strata defined by geomorphology, water characteristics, and anthropogenic use. And the graphic on the right shows the number of organisms, or total abundance, and species richness, so how many different types of species there are in these different habitat types. The habitat types on the map are the same color code as you will find in the graphic, the bars. And I want to call your attention to the pink areas. These are the harbor areas. They're very small in size, but these are areas that are usually near large river mouths, and they're important habitat for fish nurseries. 2 sampling periods are depicted. The lighter shades are for the baseline, and the darker are for the resample. And what we see is that for all the strata types, all the different habitat types, the total number of organisms has decreased between the 2 sampling periods. And the number of species decreased in all except for the deep basins of Puget Sound. So our benthic index is something that we use. It is a determination of whether a benthic assemblage is adversely affected or unaffected by any kind of stressor, whether it be anthropogenic or a natural stressor. The determination is made by benthic experts and based on calculated indices, including abundance and presence or absence of stress-sensitive species, or stress-tolerant species as well. The red indicates adversely affected benthos, both in the map and in the pie charts. And so what we see here is that when we compare the 2 sampling periods, baseline being on the left and resample on the right, is that the area with adversely affected benthos has increased from 35% to 44%. And then there is a spatial pattern as well of where those benthic communities are impaired. You will see that they're primarily in the terminal inlets of Puget Sound and Hood Canal. So, yeah, it doesn't work. So, as we've seen in the previous slides, some areas of Puget Sound have had significant declines in benthic communities. And when we look at what species are declining and what species are increasing, we see some interesting patterns. What this graphic here shows is the abundance of a small clam that is moderately tolerant to pollution, and it thrives in areas of organic enrichment. This species has increased most of all the species that we've looked at, and we have about 1,000 that we found within our program. The chart shows abundance on the y-axis in each of the regions, and the sampling periods on the x-axis. And what we see is that in Admiralty Inlet, Whidbey Basin, Central Sound, and South Sound, we've had increases of this particular animal. But we can only look at that over 10-year periods. So is this a cycle, a natural cycle that this animal is presenting, or is this an actual change over time? For that, we need to look to our long-term program. These are the 10 sites that have been sampled since 1989. This is the graphic is depicting the same small clam. And from these data, we see that the species is indeed increasing, and it is increasing exponentially in the Sound at about a rate of 10% a year. So to recap, this species has increased the most in abundance in the regions that are growing in human populations. The species does well in nutrient enrichment, aka sewage, and it has seen a systematic increase over time at all of our long-term stations. So this species may be a potential indicator of eutrophication. So to figure out why our benthic communities are declining, we look to the data that we collect along with our benthos. As I mentioned, we collect chemistry. Our chemistry is not indicating any changes in the benthos. It does not correlate well at all. And it actually shows an opposite picture. So we see decreases in detection rates. And when they are detected, they're primarily detected near urban centers. And when detected, the detection rates or the detection concentrations have gone down over time, and in some cases significantly so. And there's just no correlation between the individual chemicals or the chemicals as a whole with the benthic community. Our toxicity index is a combination of two laboratory bioassay tests. It is generally thought to pick up toxicity or synergistic effects of unmeasured chemicals. And we found that the toxicity in urban bays was less than anticipated. So usually when there's more chemicals, you find more toxicity. So near the urban bays, it was less than anticipated. And it remained statistically unchanged over the last decade. However, the spatial distribution of where we find toxicity, so that's indicated on the map by the orange and red dots, is primarily in the terminal inlets of Puget Sound. So the same places where we have issues with our benthos. However, toxicity results only weakly correlate with our benthic communities. So the summary of findings from our program in general terms is that the benthos is declining over time. Benthos of adversely affected communities are usually in terminal inlets. And the increase of pollution-tolerant species could potentially be an indicator. Higher toxicity is also found in terminal inlets, and the laboratory chemistry and toxicity tests do not correlate well with our benthos. So there's no smoking gun. So we don't have all the answers. Our supporting data is not giving us any indication of why our benthos are declining. And so we look to other programs and come up with other hypotheses, Christopher's being one of them. We also look to where we can mine other data, and I'm going to show you some examples of where we're taking the model and lining that up with our benthos. So what we're looking at here is a model output from 2006 of DO depletion in Puget Sound. And the depletion is thought to be from human nutrient load, so it's not anthropogenic, or it's not natural, it's anthropogenic. The figure here shows the colors would be, the blue would be good oxygen, and the yellows and oranges would be degraded oxygen. And what we see is that the largest impact in Puget Sound are in Whidbey Basin, southern Skagit Bay, Port Susan, Bainbridge Basin, South Sound, and Hood Canal. Again, some of the same areas where we have seen benthic impairment. I want to make some comparisons between two model outputs. On the left here is the 2006 model output, and on the right is the 2008. These 2 years were very different hydrologically. 2006 had much higher river flows, or much lower river flows, and 2008 had much higher river flows. So note the areas in pink, which are all areas where water exchange is naturally slow due to the bathymetry and the shape of the sound. But note how the magnitude and spatial extent of DO in 2006 is greater than in 2008. So keeping in mind those model outputs from the previous slide, we want to look at residence time. The years were different hydrologically. 2006 had much lower river flows, which translated into longer residence time in the sound, and 2008 had the higher flows, which translated into lower residence time. So if you remember from the maps, the DO depletion was greater in magnitude and spatial extent when we had the lower river flows. So one link between residence time, productivity of organic matter, and benthic communities is dissolved oxygen. In very general terms, Puget Sound has high productivity in the spring and summer months, with a plankton die-off later in the fall, drawing down DO by the decomposition of the leftover organic matter. The map shows the 2006 model output, again of the low DO, that occurred in the Salish Sea. The greens are higher DOs, moving to the lower in the yellows, oranges, and reds. I've plotted our benthic impaired stations as red dots and the unimpaired stations as green dots. And what you want to do is you want to match up where the reds coincide. Again, those are in the terminal inlets, southern Hood Canal, up in Bellingham Bay and Skagit. So we wanted to see if there was concordance. Visually, yes, there is, but we also wanted to make sure that it wasn't just an optical illusion. So I took the data from the model, and since I overlaid our benthos, I could see how many of our impaired stations actually fell in impaired DO waters. And what you can see from the graphic on the right is that the greater percentage of impaired sites actually occur where there's lower DO levels. So there is good concordance. All right, how productive a system is important as well. So how much organic matter occurs. Ecology implemented a particulate pilot project to look at organic matter in the form of carbon and nitrogen, to monitor the changes in spatial and temporal patterns in the material that's in the water column at the surface, and eventually making it down to the sediment. What we're looking at here is early results of this particulate project, so since about April of 2016, it's about a year's worth. And what we see is that the organic nitrogen has a lot of variability in it. There's a lot of seasonal variability, but some interesting things pop out. Some areas have higher productivity, so where the bars are taller. The orange is for the 10 meters, and the green is for the near bottom. And so we see that the bars, the general patterns line up. If you have higher productivity in the surface, you will have more of the particulate matter coming down to the bottom. So we see that some of the areas in pink here are areas, Budd Inlet, Case Inlet, Hood Canal, Sinclair, and the the Skagit, are all areas, again, where we've seen this benthic impairment. They're really highly productive, there's slow flushing, and a lot of primary productivity. This primary productivity is also documented by the Eyes Over the Puget Sound flight program, where they document areas that have large blooms during the summers, especially the dinoflagella blooms, and they also have large presence of jellyfish, both which have been documented to be eutrophication indicators. So there's lines of evidence that something is going on. More nutrients, slower flushing could potentially cause some big problems. So what does it mean for the future? Areas with sluggish water exchange will likely increase if we continue to have climate change and have our flushing not be as good. Areas with low oxygen zones will likely increase, and biogeochemical processes will change along with that. The amounts of nutrients released to the water column will most likely change, and areas with adversely affected benthos are likely to increase as well, which of course will cause issues for food webs. Additional nutrient loads and climate change have large implications for Puget Sound, not just for benthic communities, but just about every interaction and process. Changes in flushing rates, nutrient cycles, and the condition of the benthos are sure to be impacted. So knowing this, where do we intend to move into the future? We will continue to monitor benthic invertebrate communities and start to look at the zooplankton communities as well. What is transferring up? Are those species changing? And what impacts could they potentially have on the food web? We will monitor counts, size classes, and biomass for both. And we will continue to monitor water column particulates both at the surface and at the bottom. Sediment chemistry will be continued and will be expanded to tissue chemistry in the benthos and in zooplankton for priority pollutant metals, organics, and chemicals of emerging concern. The program has already added parameters to get a better handle at the biogeochemistry, including total carbon, total inorganic carbon, total nitrogen, and carbon to nitrogen ratios. We have just finished conducting a pilot study up in Bellingham Bay looking at fluxes, sediment fluxes, so how are nutrients transferring back to the water column? And we hope to continue that as well. But of course, all of this work is dependent on approved budgets. So with that, I will take questions.
Do you have any questions out there? Nailed it. Oh, maybe we have one here. Yeah, that's it. Back to the mic.
Might take a second. There we go. The biovalve that you showed, the parvalucina, I believe it was, that is a taxa that's tolerant of low dissolved oxygen conditions. Is that correct?
No, they're they're moderately tolerant to pollution and like areas of organic enrichment. So near sewage outfalls, they do really well. Okay. So we're seeing the increase in spatial extent of that particular taxa, as well as over time.
Are there other taxa that could also be good indicator species to focus on?
Yes, we have several. I just chose one.
Okay. Thanks.
Okay. We've got a question over here. Lincoln?
Going back to that same species that you said is more pollutant tolerant, is it, do you have indications of increased organic enrichment in those same sediments where it's decreasing? Because I would think it may be going the other way.
So we have not monitored for organic enrichment with the exception of the particulate pilot study, and that was only for a year. So this species has increased over the last 20 years. So we don't have the data to look at that.
Yeah, because the organic particulate loading has been going down with advanced treatment. And I'm just kind of wondering now how this all works.
We see it increasing in areas where population is really growing, particularly South Sound, Central Sound, and then Skagit area. So, like I said, we don't have the data to actually